Initially, this transforming principle, what Griffith was thinking, scientists probably thought that this transforming principle might be a protein. But in the year 1934, two biologists, Avery and MacLeod, extrapolated the experiments of Griffith worked on the same bacteria, Streptococcus pneumoniae, and found that the transforming principle was DNA and not any protein. So from now onwards, scientists concentrated on whether DNA is the real transforming principle, whether DNA is the one that is functioning as the genetic material. It was in 1952, two scientists, Hershey and Chase, working on bacteriophages, very clearly established that the transforming principle was DNA and not the protein code. They used radioactivity to conduct this experiment. They took radioactively labeled sulfur and nitrogen. So in their experiments, Hershey and Chase, they used bacteriophages and infection of these bacteriophages into E. coli bacteria, Escherichia coli bacteria. They took some E. coli bacteria that were labeled with radioactive sulfur 35S, some with 32 phosphorus, and found that the phages multiplied in both the cases. Subsequently, they separated the protein coat and the viral particle by blending and centrifugation. Labeled the protein coat with radioactive sulfur, labeled the viral particle containing DNA with radioactive phosphorus. So phage with radioactive protein coat was made to infect the bacterial cell. The phage entered into the non-radioactive bacteria, multiplied vigorously. Phage multiplied, but the bacteria remained non-radioactive. In the other case, phage with radioactive DNA, no nitrogen in the protein. Phage was infected into E. coli bacteria. Phage multiplied vigorously. Phage multiplied vigorously, and the bacteria became radioactive. That means here radioactivity was not detected in the E. coli cells when only the protein coat of the phage was made radioactive, but the bacteria became radioactive when the phage DNA was radioactive, very clearly indicating that the transforming principle was DNA and not any protein. So this experiment very clearly established the fact that it is the DNA that serves as the genetic material that is involved in transformation, and that is the one which is transferred from cell to cell.